Welcome to another deep dive, everyone. Today, we're going to be tackling something pretty complex, but also super important, carbohydrate metabolism. And get this, we're going all the way down to the molecular level.
It's going to be a wild ride.
You said it. Now, this deep dive is especially useful for all you second-year biology and biotechnology students out there.
But really, anyone who's ever wondered how their body actually works at a molecular level will find this fascinating.
Totally. I mean, who doesn't want to know how their body turns food into energy, right?
Right. And today we're focusing on two key players in that process. Gluconeogenesis.
Say that again?
Gluconeogenesis.
Gluconeogenesis. Okay, I think I got it.
And glycogen metabolism. These pathways are essential for keeping our energy levels balanced, making sure our bodies have glucose when they need it.
Glucose.
Yeah.
That's our primary energy source, right? Kind of like the fuel that keeps our cells running.
Exactly. And gluconeogenesis is like our backup generator, kicking in when we're low on glucose from food. It's how our bodies make glucose from scratch, using other molecules like lactate or amino acids.
Wait, so we can actually make glucose from things besides just the carbs we eat?
Yep. Pretty cool, huh? It's a lifesaver when our blood sugar dips too low.
That's amazing. Okay, I'm starting to see why this is so important. But before we get too deep into gluconeogenesis, let's do a quick recap of glycolysis.
Oh yeah, glycolysis, that's where it all begins.
Right. Glycolysis is the breakdown of glucose to produce energy. It's like the first step in getting ATP, the energy currency of our cells.
And a pretty efficient one too. Glycolysis nets us 2 ATP molecules per glucose molecule, along with 2 pyruvate molecules.
Okay, so we get some energy and these pyruvate molecules. What happens to them?
Well, those pyruvates can go a couple of different routes. Under normal conditions, with plenty of oxygen around, they head into the mitochondria, the powerhouse of the cell, and get further oxidized in the citric acid cycle to generate even more ATP.
Got it. So that's like the main energy production pathway. But what about when oxygen is limited, like during intense exercise?
That's when things get interesting. Under anaerobic conditions, those pyruvates can be converted to lactate through fermentation. This process regenerates NAD plus Benc, which is essential for keeping glycolysis going.
Okay, so fermentation is like a temporary fix, keeping glycolysis running when we don't have enough oxygen.
Exactly. It's like a quick energy boost, but it can't sustain us for long.
Makes sense. Now, how does all of this tie back into gluconeogenesis?
Well, remember those pyruvate molecules we talked about? They're not just destined for the citric acid cycle or fermentation. They can also be used as a starting point for gluconeogenesis.
I see. So it's like those pyruvates are at a crossroads. They can go down the energy production route or be used to make glucose through gluconeogenesis.
Exactly. And that decision depends on the body's energy needs. When glucose is scarce, those pyruvates get diverted towards gluconeogenesis.
Okay, that makes sense. But hold on, I thought gluconeogenesis was just glycolysis in reverse. Is it really that simple?
That's a common misconception. While gluconeogenesis does share some reversible reactions with glycolysis, it's not just a simple rewind. There are some crucial differences in the pathways.
Okay, so it's more like a remix than a straight-up reversal.
Yeah, that's a good analogy. There are some key detours and modifications that make gluconeogenesis a unique pathway in its own right.
Okay, I'm intrigued. Let's dive into those differences. What are the key steps that set gluconeogenesis apart from glycolysis?
The most important differences are 3 irreversible steps in glycolysis that have to be bypassed in gluconeogenesis.
Irreversible steps, meaning they can't just run in reverse.
Exactly. These steps require specific enzymes that are unique to gluconeogenesis, allowing the pathway to move forward despite those roadblocks in glycolysis.
Okay, I'm starting to see how this works. So let's break down those bypasses one by one. Where's the first roadblock we encounter in gluconeogenesis?
The first one is right at the beginning, converting pyruvate back to phosphenolpyruvate, or PEP for short.
TP. That's one of the intermediates in glycolysis, right?
Yeah. And getting back to PP from pyruvate is a 2-step process, and it requires a bit of an energy investment.
Sounds intriguing. What are those 2 steps?
First, pyruvate gets transported into the mitochondria, where it's converted to oxaloacetate by an enzyme called pyruvate carboxylase.
Pyruvate carboxylase. Okay, and what does this enzyme need to do its job?
It needs ATP, our cellular energy currency. So right off the bat, gluconeogenesis is costing us energy.
Interesting. So even though we're making our own glucose, it's not a free lunch.
Nope. Then that oxaloacetate gets shuttled back out to the cytoplasm, where it's converted to PP by another enzyme called phosphenolpyruvate carboxykinase, or PPCK for short.
TPCK. Wow. These enzyme names are a mouthful.
I know, right?
Oh.
But they do important work, and PPCK needs GTP, another energy carrier, to do its job.
So we've already spent 2 energy molecules just to get back to PP.
Precisely. Gluconeogenesis is an energy-intensive process, but it's essential for keeping our glucose levels stable when we're not getting enough from food.
Okay, I'm starting to appreciate the complexities here. So we've bypassed the first irreversible step in glycolysis. What's next?
The second bypass involves converting fructose 1,6-bisphosphate to fructose 6-phosphate.
Fructose 1,56-bisphosphate, another familiar name from glycolysis.
Yep, and this conversion is catalyzed by an enzyme called fructose 1,6-bisphosphatase, or FBPase1 for short.
FBPase1, got it. And this step doesn't require any extra energy, right?
Nope, this one's a simple hydrolysis reaction. The enzyme just removes a phosphate group from fructose 1,6-bisphosphate to make fructose 6-phosphate.
Okay, that's a bit less energy demanding.
Yeah.
So what about the final bypass?
The final bypass is the conversion of glucose 6-phosphate to glucose, which happens in the endoplasmic reticulum.
The endoplasmic reticulum, that's a different cellular compartment. Why does it happen there?
Good question. The enzyme that catalyzes this step, glucose 6-phosphatase, is membrane-bound and located in the endoplasmic reticulum.
Okay, so it's a location-specific reaction. And what does glucose 6-phosphatase do?
It simply removes a phosphate group from glucose 6-phosphate, releasing free glucose.
Free glucose. That's what we're after, right? The glucose that can be used by our cells or released into the bloodstream.
Exactly. So we've bypassed all 3 irreversible steps in glycolysis, and we've successfully synthesized glucose from pyruvate.
This is incredible. It's like a whole new pathway built to work around those roadblocks in glycolysis.
It is, and it highlights how adaptable and resourceful our bodies are when it comes to maintaining glucose homeostasis.
Homeostasis, that's the key, right? Keeping our blood sugar levels stable even when we're not constantly eating.
Exactly. Gluconeogenesis is a critical part of that balance, ensuring a steady supply of glucose to our brains and other vital organs.
Okay, I think I'm starting to wrap my head around gluconeogenesis now. It's like our body's way of saying, nope, not today, low blood sugar.
Exactly. Our bodies are pretty clever when it comes to keeping things running smoothly.
But you know, it's not lost on me that we've talked a lot about making glucose. But what about storing it?
You're talking about glycogen. That's our glucose savings account, allows us to tuck away excess glucose when we've got plenty.
And then withdraw it when we need a quick energy fix.
Right. And just like a real bank account, the balance of our glycogen stores is carefully managed.
It sounds like a pretty important job. So tell me more about glycogen. Like, what is it exactly?
Well, glycogen is basically a big branched chain of glucose molecules all linked together. It's the way our bodies store glucose for later use.
Okay, so it's like a glucose warehouse, all neatly organized and ready to go.
Yeah, something like that. And we've got these glycogen stores mainly in our liver and skeletal muscles.
Liver and muscles. Makes sense. Those are pretty energy-hungry tissues.
Exactly. And they actually use glycogen for slightly different purposes.
Oh. Tell me more.
Well, in muscle, glycogen is mainly an energy reserve for, muscle contraction. Like when you're sprinting or lifting weights, your muscles tap into those glycogen stores for fuel.
Okay, so it's like a local energy source for those quick bursts of activity.
Right. But liver glycogen, that's got a slightly different role. It's more about keeping our blood sugar levels stable. Like when we haven't eaten for a while, the liver breaks down glycogen and releases glucose into the bloodstream.
So it's like the liver is the generous friend sharing its glucose with everyone else.
Exactly. It's all about maintaining balance and making sure all our tissues have the glucose they need to function.
This is so fascinating. It's like our bodies have this whole internal economy going on, managing resources and making sure everything runs smoothly.
It really is. And all of this is happening at a molecular level, which is pretty mind-blowing when you think about it.
Okay, so we've got the what of glycogen, but I'm really curious about the how. Like, how do our bodies actually break down and build up these glycogen stores?
Great question. Let's start with glycogen breakdown, also called glycogenolysis. It's basically a multi-step process that starts with an enzyme called glycogen phosphorylase.
Glycogen phosphorylase. Catchy name. What does it do?
It's the key player in breaking down glycogen. It cleaves off glucose units from the ends of those glycogen chains, releasing glucose 1-phosphate.
Glucose 1-phosphate, so not just plain glucose.
Right. It's glucose with a phosphate group attached. This phosphate group is important because it helps trap the glucose inside the cell and keeps it from just diffusing out.
Okay, that makes sense. So glycogen phosphorylase is like the demolition crew chipping away at the glycogen molecule.
Yeah, good analogy. But it can't handle everything on its own. Remember those branches in the glycogen molecule? Well, glycogen phosphorylase can't break those down.
So we need a specialist for that.
You got it. We've got another enzyme called the debranching enzyme, and it comes in and handles those branch points. First, it shifts a few glucose units from the branch to a nearby chain, and then it cleaves off the remaining glucose, releasing free glucose.
So it's like a two-step process. The debranching enzyme is like a tree trimmer, making sure all those branches are properly pruned.
Exactly. Teamwork at its finest, right?
Totally. Now, what happens to all that glucose that's been released from glycogen?
Well, the glucose 1-phosphate produced by glycogen phosphorylase gets converted to glucose 6-phosphate by another enzyme.
And then?
Then it depends on where we are in the body. In muscle, that glucose 6-phosphate usually enters glycolysis, providing fuel for muscle contraction.
Makes sense. Going to keep those muscles energized. But what about in the liver?
Ah, the liver. Remember its role in maintaining blood sugar. Well, in the liver, glucose 6-phosphate can be converted to free glucose by another enzyme and then released into the bloodstream.
So it's like the liver is a glucose distribution center, sending out supplies to whoever needs them.
Exactly. It's all about keeping those glucose levels balanced no matter what's going on in the body.
This is amazing. But we've only talked about breaking down glycogen. How do we build it back up? I mean, got to replenish those energy reserves, right?
Absolutely. That process is called glycogen synthesis or glycogenesis, and it's essentially the reverse of glycogenolysis, but with a few key twists.
Twists? Like what?
Well, for starters, instead of using free glucose, glycogen synthesis uses an activated form of glucose called UDP glucose.
UDP glucose. Never heard of it.
It stands for uridine diphosphate glucose, but you can just call it UDP glucose. It's basically glucose with a high-energy tag attached, making it ready for action.
Okay, so it's like glucose with a VIP pass, getting priority access to glycogen synthesis.
Exactly. And the enzyme that uses this VIP glucose is called glycogen synthase.
Glycogen synthase. I'm sensing a pattern here. Breakdown has phosphorylase and buildup has synthase.
You're catching on. And glycogen synthase is the key player in building up those glycogen chains. It adds glucose units from UDP glucose to the growing glycogen molecule.
Okay, so glycogen synthase is the construction crew, putting those glucose bricks together.
I like it. But remember those branches? Well, glycogen synthase can only make straight chains, so we need another enzyme, the branching enzyme, to create those branches.
Or branching enzyme. I should have guessed. So it's kind of like an architect making sure the glycogen structure is nice and complex.
Exactly. And all of this synthesis and breakdown is happening constantly in our bodies, carefully regulated to match our energy needs.
It really is incredible. But speaking of regulation, I'm kind of curious how all of this is controlled. I mean, we've got these opposing processes happening, breakdown and synthesis. So how do our bodies avoid creating a chaotic mess?
That's where things get really interesting. Glycogen metabolism is tightly regulated by a combination of allosteric effectors and hormonal signals.
Allosteric effectors and hormones. Okay, I'm listening.
So remember our old friend fructose 2-100.6 bisphosphate? It's not just involved in glycolysis and gluconeogenesis.
Wait, it plays a role in glycogen metabolism too. This molecule is a real multitasker.
You bet. And the key here is that high levels of fructose 200.6 bisphosphate stimulate glycolysis and indirectly promote glycogen synthesis. Remember, those high levels are typically triggered by insulin, the hormone released when blood sugar is high.
Right. So when we've just eaten and our blood sugar's up, insulin tells our bodies to store some of that glucose as glycogen.
Exactly. Insulin indirectly activates an enzyme called phosphoprotein phosphatase 1, or PP1 for short. And PP1 dephosphorylates glycogen synthase, making it more active.
So insulin, through PP1, essentially flips the switch towards glycogen storage when glucose is abundant.
Exactly. And on the flip side, when blood glucose levels drop, we've got glucagon stepping in.
Glucagon, the counterpoint to insulin.
Yeah.
What does it do in this scenario?
Glucagon triggers a cascade of events that ultimately leads to the phosphorylation and activation of glycogen phosphorylase. Remember, that's our glucose-releasing enzyme.
So glucagon puts the brakes on glycogen synthesis and activates glycogen breakdown, making sure we have glucose available when we need it.
Precisely. And on top of that, glycogen phosphorylase is also allosterically regulated. For example, in muscle, it's activated by AMP, a signal of low energy. Basically, when muscle cells are working hard and ATP levels are low, AMP builds up and tells glycogen phosphorylase to release more glucose for fuel.
So it's like the muscle saying, hey, I need more energy here, break down some glycogen stat.
Exactly. It's a really efficient way to adjust energy production to meet demand.
This whole system of regulation is just mind-blowing. It's like a perfectly choreographed dance, ensuring our bodies are always in balance.
I know, right? And it highlights just how important these pathways are for human health. When these finely tuned systems go wrong, it can lead to some serious problems.
Like what?
Well, there are a number of genetic disorders that can disrupt glycogen metabolism, leading to what we call glycogen storage diseases.
Glycogen storage diseases. What are those like?
These are conditions where specific enzymes involved in glycogen synthesis or breakdown are deficient or malfunctioning. This can lead to the accumulation of glycogen in certain tissues, causing all sorts of problems.
Are there any specific examples you can give?
Sure. One classic example is von Gierk's disease. This is caused by a deficiency in the enzyme glucose-6-phosphatase. Remember, that's the enzyme that converts glucose-6-phosphate to glucose in the liver, allowing the liver to release glucose into the bloodstream.
Right. We talked about that in the context of gluconeogenesis, too.
Exactly. So in people with von Gierke's disease, the liver can't release glucose properly, leading to severe hypoglycemia, which is when blood sugar levels get dangerously low.
Wow, that sounds pretty serious.
It is, and it really highlights how even a single enzyme deficiency can have a huge impact on overall health.
It makes you appreciate just how intricate and interconnected these pathways are. Everything has to work together perfectly to keep us functioning properly.
Right. It's all about balance and coordination. But speaking of interconnected pathways, there's another one we need to touch on that's closely related to glucose metabolism.
I'm intrigued. What is it?
It's called the pentose phosphate pathway, or PPP for short. It's like a side branch off of glycolysis, and it plays a crucial role in providing the building blocks for other essential molecules and protecting cells from damage.
Okay, so not directly involved in energy production, but still super important.
Exactly. It's like a support team, making sure everything else runs smoothly. The PPP produces two main products, NADPH and pentose sugars.
NADPH. That sounds familiar. It's related to NAD plus XC, right? That molecule that's essential for glycolysis in the citric acid cycle.
Yep. NADPH is the reduced form of NADP plus XC, and it's a powerful reducing agent, meaning it donates electrons readily. This makes it essential for various biosynthetic reactions, like making fatty acids and cholesterol.
So the PPP provides the reducing power needed to build these important molecules.
Precisely. And NADPH is also crucial for protecting cells from oxidative damage.
Oxidative damage, what's that?
Our cells are constantly exposed to reactive oxygen species, or ROS, which are basically byproducts of normal metabolism. But these ROS can be pretty harmful, damaging DNA, proteins, and even the cell membrane.
So they're like little cellular troublemakers.
You could say that, but NADPH helps to neutralize these ROS, acting like a cellular antioxidant. It's like a defense system, protecting our cells from these harmful molecules.
Wow, so the PPP is involved in both building essential molecules and protecting our cells from harm. That's pretty impressive for a side branch pathway.
Yeah, it is. It just shows you how everything is connected and how our bodies have evolved these intricate mechanisms to keep everything running smoothly.
We can't forget about those pentose sugars, right?
Oh, yeah, those are important too. Pentose sugars, like ribose 5-phosphate, are essential components of nucleotides.
Nucleotides, the building blocks of DNA and RNA? So the PPP is providing the raw materials for making our genetic material.
Exactly. Without the PPP, our cells wouldn't be able to replicate their DNA or synthesize RNA, which are essential for protein synthesis and countless other cellular processes.
This is incredible. It's amazing how this seemingly minor pathway plays such a major role in so many critical functions.
It's a testament to the interconnectedness and efficiency of metabolic pathways. Everything is linked and regulated to ensure our cells have the resources they need to function properly.
And this regulation extends to the PPP as well, right?
Absolutely. The main regulatory point of the PPP is the first enzyme in the pathway, glucose-6-phosphate dehydrogenase, or G6PD for short.
G6PD, another enzyme with a long name. So how is it regulated?
The activity of G6PD is mainly controlled by the level of NADP plus the oxidized form of NADPH. Basically, when NADPH levels are high, G6PD activity is inhibited because the cell already has enough NADPH.
Right. It's like a feedback loop, making sure the cell doesn't overproduce NADPH.
Exactly. And conversely, when NADPH levels are low, G6PD activity is stimulated to make more.
So it's all about maintaining that balance again, ensuring a steady supply of NADPH without going overboard.
Precisely. It's a beautifully elegant system of regulation.
This has been such an enlightening discussion. We've gone from the basics of glycolysis all the way to the pentose phosphate pathway, and it's clear that these pathways are incredibly complex and interconnected.
It really highlights the fascinating world of biochemistry and how much is happening within our cells every second of every day.
And speaking of our deep dive, let's bring it back to our focus for today, which is specifically looking at the biochemical details of gluconeogenesis and glycogen metabolism. This is particularly relevant for all you second-year biology and biotechnology students out there.
Absolutely. These pathways are fundamental to understanding human metabolism, and they're often covered in detail in your coursework. So let's hit some key points that are particularly important for your studies.
Sounds good. Let's start with gluconeogenesis. One thing that really stood out to me is the energetic cost of this pathway. I mean, it takes a lot of energy to build glucose from scratch. Remember all those ATP and GTP molecules we talked about?
Right. To be precise, it takes 6 ATP, 2 GTP, and 2 NADH molecules to synthesize just 1 molecule of glucose from 2 molecules of pyruvate. That's a significant energy investment.
Yeah, it's definitely not a free lunch. So why do our bodies even bother with this seemingly expensive pathway?
It all boils down to the critical importance of glucose for certain tissues, particularly the brain. The brain has a very high metabolic demand and relies heavily on glucose as its primary fuel source. It can use other energy sources like ketone bodies, but glucose is its preferred fuel.
So it's a matter of survival. Our bodies are willing to pay a premium to ensure a constant supply of glucose to the brain, especially when dietary sources are scarce.
Exactly. The brain's need for glucose outweighs the energetic expense of gluconeogenesis. It's a trade-off that evolution has favored to ensure our cognitive function and survival. I mean, without a functioning brain, we wouldn't be here having this conversation.
Very true. And this brings us back to the elegant interplay of these pathways. When glucose is plentiful, like after a meal, our bodies favor glycolysis and glycogen synthesis, using that glucose for energy and storing the excess as glycogen.
But when blood glucose levels drop, gluconeogenesis and glycogen breakdown kick in, making sure those glucose-dependent tissues, like the brain, have a constant supply. It's like our bodies have a backup generator, always ready to switch on when the main power supply falters.
A very sophisticated backup generator. It's amazing how our bodies can sense those changes in blood sugar and adjust these pathways accordingly.
It really is. And it all comes down to those enzymes and hormones we've been talking about working together to keep everything in balance.
Now let's shift gears and revisit glycogen metabolism. One thing that often trips students up is the different roles of glycogen in muscle and liver. We touched on them earlier, but let's break it down further.
Okay, good idea. So both muscle and liver store glycogen, but they use it for different purposes. Muscle glycogen is like a local energy reserve, specifically for powering muscle contraction. It's selfish, only used by the muscle itself.
And the liver is more like a communal pantry, sharing its glycogen stores with the whole body.
Exactly. The liver breaks down glycogen and releases glucose into the bloodstream to maintain blood glucose levels, ensuring a constant supply to those glucose-dependent tissues.
Okay, so the purpose is clear. But what's the actual difference at a biochemical level? I mean, glycogen is glycogen, right?
Well, structurally, glycogen is the same in both tissues, but there's a key enzymatic difference that explains their distinct functions.
Okay, I'm all ears. Tell me about this enzymatic difference.
It comes down to the enzyme glucose-6-phosphatase. Remember, that's the enzyme that converts glucose-6-phosphate to glucose, the final step in both gluconeogenesis and glycogen breakdown in the liver. It's what allows the liver to release glucose into the bloodstream.
Right, but you said there's a difference, so what's the catch?
The catch is that muscle cells don't express glucose-6-phosphatase.
Ah, so they can break down glycogen to glucose-6-phosphate, but they can't convert it to free glucose.
Exactly. This means that muscle glycogen is strictly for internal use by the muscle itself. It can't contribute to blood glucose levels.
That makes perfect sense now. It's all about which enzymes are present in each tissue. It's a beautiful example of how subtle differences at a molecular level can have huge functional consequences.
It really is. It highlights the importance of understanding the details, the specific enzymes, and their regulation to fully grasp how these pathways work.
This has been such an amazing journey through carbohydrate metabolism. I feel like I've gained a whole new level of understanding of these pathways and how they work together to keep our bodies running.
Me too. It's a testament to the power of biochemistry, uncovering the molecular mechanisms that underpin life itself.
And remember, we've only just scratched the surface. There's so much more to explore, so much more to learn about these intricate pathways and how they're integrated with other cellular processes.
Exactly. So keep those minds curious, keep asking questions, and keep delving deeper into this fascinating world. The journey of discovery in carbohydrate metabolism is just beginning.
Thanks for joining us on this deep dive into carbohydrate metabolism. We hope you've enjoyed exploring the intricate world of gluconeogenesis, glycogen metabolism, and the pentose phosphate pathway. Until next time, stay curious. So where do we go from here? We've covered so much ground, from glycolysis to gluconeogenesis to glycogen metabolism, even that pentose phosphate pathway.
It's been quite a journey through the world of carbohydrates, hasn't it?
Yeah, and I have to say, I'm really starting to appreciate just how complex and interconnected all these pathways are. It's like a finely tuned machine with each part playing a critical role.
Exactly. And it all comes back to that fundamental concept of balance. Our bodies are constantly striving to maintain homeostasis, keeping everything within a narrow range, whether it's blood sugar levels, energy production, or even the synthesis of essential molecules.
And that balance is achieved through this intricate network of metabolic pathways, all carefully regulated by enzymes, hormones, and even those allosteric effectors we talked about.
It's really amazing when you think about it. All of this is happening within our cells every second of every day without us even being aware of it. Just a symphony of biochemical reactions, all working together to keep us alive and functioning.
Okay, so before we get too philosophical here, let's bring it back to our focus for this deep dive. Gluconeogenesis and glycogen metabolism. These are the pathways that are particularly relevant to you as a second-year biology and biotechnology student, right?
Absolutely. These are fundamental concepts that you'll encounter again and again in your studies and research. And having a solid understanding of these pathways will really give you a leg up in your coursework and future careers.
So let's do a quick recap of those key takeaways that you really need to remember about gluconeogenesis and glycogen metabolism. Sound good?
Sounds great. Let's start with gluconeogenesis, that remarkable pathway that allows us to make glucose from scratch. The first thing to remember is that it's an energy-intensive process. It takes 6 ATP, 2 GTP, and 2 NADH molecules to synthesize 1 glucose molecule from 2 pyruvates. That's a lot of energy.
Yeah, we've said it before, but it's worth repeating. Making glucose isn't cheap. So why do our bodies even bother with this seemingly expensive pathway? We talked about this earlier, but it's a key point, so let's recap it again.
The reason is simple: survival. Glucose is essential for certain tissues, particularly the brain. The brain has a high metabolic demand and relies heavily on glucose as its primary fuel source. It can use other energy sources, but glucose is its preferred choice.
So our bodies are willing to pay a premium, energy-wise, to keep the brain happy.
Precisely. The brain's need for glucose trumps the energetic cost of gluconeogenesis. It's a trade-off that evolution has favored to ensure our cognitive function and survival. I mean, think about it. Without a functioning brain, well, we wouldn't be here to worry about gluconeogenesis in the first place.
Good point. Okay, so gluconeogenesis is essential for keeping our brains powered up. But what about glycogen metabolism? How does that fit into the overall picture of energy balance?
Glycogen metabolism is all about storage and retrieval. When we've got plenty of glucose, like after a meal, our bodies store the excess as glycogen, mainly in the liver and muscles.
And then when blood sugar levels drop, those glycogen stores are broken down to release glucose, keeping our energy levels stable.
Exactly. It's like having a backup generator, always ready to kick in when the main power supply falters. And just like with gluconeogenesis, glycogen metabolism is tightly regulated, responding to hormonal signals and energy demands.
It's all about balance, right? Making sure our bodies have the right amount of glucose at the right time, whether it's coming from food, gluconeogenesis, or those glycogen stores.
Balance is key. And that's one of the most important takeaways from this whole discussion. Our bodies are constantly striving to maintain homeostasis, and these metabolic pathways are a critical part of that balancing act.
Now, I remember we talked about how glycogen plays different roles in muscle and liver. Can you remind me what those differences are?
Sure, both muscle and liver store glycogen, but they use it for different purposes. Muscle glycogen is like a local energy reserve, exclusively for powering muscle contraction. Think of it as a selfish hoarder, only sharing with the muscle fibers themselves. On the other hand, liver glycogen is more altruistic, sharing its glucose with the entire body. It's broken down to release glucose into the bloodstream, keeping those glucose-dependent tissues like the brain happy and fed.
It's fascinating how the same molecule can have such distinct roles depending on where it's stored.
Right, and it all boils down to a single enzyme, glucose 6-phosphatase. Liver cells express this enzyme, allowing them to release free glucose into the bloodstream. Muscle cells, however, lack this enzyme, so they're stuck with glucose 6-phosphate, which can only be used internally by the muscle itself.
It's a prime example of how subtle differences in enzyme expression can lead to major functional differences between tissues.
It really shows you how important it is to understand the details, the specific enzymes and their regulation, to truly grasp how these pathways operate.
This has been such an incredible deep dive. We've explored gluconeogenesis, glycogen metabolism, and even touched on the pentose phosphate pathway. And it's clear that these pathways are not only complex, but also incredibly interconnected, working together to orchestrate the symphony of carbohydrate metabolism.
And remember, we've only just scratched the surface. There's still so much more to learn about these pathways and how they interact with other cellular processes. So keep those minds curious, keep asking questions, and keep exploring this fascinating world within.
Well, that about wraps up our deep dive into carbohydrate metabolism. We hope you've enjoyed the journey and learned some valuable insights along the way.
And for all you aspiring biologists and biotechnologists out there, remember that the journey of discovery is just beginning. Keep those minds engaged, keep learning, and keep pushing the boundaries of knowledge. Who knows what amazing discoveries await?
Thanks for joining us. Until next time, stay curious.